So when it comes to validation, as far as sterilization is concerned, the machine is very important. You have to standardize the machine and check that your machine is working well. The vacuum part is very important, the temperature and the TST, that is temperature, saturation and the time. And we have different indicators. The best one is the biological indicator. Then I showed you the ultrasonic cleaner and documentation. Above all, you will have to review your performance and the entire process periodically. So the indicators are classified into class 1 to class 6. We use the class 1, class 2, 3, 4 is usually used in industrial purposes like in chocolate factory, food factories and all. 5 and 6 again we use. I'll tell you. This is class 1. Class 1, what does it say? That particular set has gone into autoclave and come out, or autoclave or sterilization is, it doesn't say whether it is perfect or not. So it is usually used to stick on top of the pack like this. Okay. So that is class 1. Class 2 is Bovidic. How Bovidic is done is, the Bovidic sheet is kept between sheets like this, packed like this, and in an empty load. No other load should be there, only these packs should be done. It is usually done immediately before the sterilization happens. And the color change should be uniform like this and documented to prove that your autoclave machine is working. And these are the different models and designs which is available commercially. And these things, and class 6 indicator can be used in the helix tube. This is helix tube is to check the vacuum of your autoclave. This is a tube which is about 1.5 meters long with 1.5 diameter inner lumen. The indicator is placed in the end of it and it's sealed airtight. And this is placed in between the load what you are doing and checked for autoclaving. This will tell you whether the machine has got good vacuum cycle. So once it is ready, you check the class 1 is checked, the helix tube is taken out and check for the indicator. This is the class 5 indicator which I will be showing you again. This is the helix tube indicator. And if there is leak or the vacuum is not clear, the color remains pink. Okay. And this is the best helix tube available commercially, which is made of metal. It's got a long tube, as I told you. That part of it is silicon, and the airtight component in this is because of there is a seal there. A rubber seal is there which will give you. This is what is recommended, but the plastic ones are also available. And what I am showing here is a batch of 10 helix tube. These tubes are used to standardize your autoclave. This is not done by you, but by the company when they come to install. This 10 helix tube are of different size and calibers. All the 10 has to pass the test if your autoclave is good. And this is called the process challenging device. This is cut open to show you that how it looks inside. Actually, it will be fully covered. You will not be able to see these tubes and all like what you see in the green part. The open part is this one. This different color is for different specialty. The yellow is for the dental, the white is for general surgery, green is for ophthalmology, because the length of the inner tube, that steel tube, is different. So it helps us to, this again tells us the vacuum cycle in the autoclave. And you can use biological indicator also in process challenging device, PCD it is called. Okay. And this is class 5. The disadvantage with this is, see it says pass, fail. If the black passes in between, pass partially, we still say it is passed. But class 6 is, it abruptly changes from yellow to blue once the autoclave process is over. So class 6 is little better than this is a dual indicator, whether your sterilization occurred is within 3 minutes or 10 minutes. So it's a dual thing. These are the different class 6 emulators available in the market, and the color change also is. Now I'll show you how to place an indicator in the set. That is class 5 within the instrument tray along with the instruments. Seal it. Biological indicators is the best for checking your sterilization. Contains 1 million spores within it, and there are different biological indicator for steam as well as the ETO sterilization. Now this is the way it is done. It is kept within the batch along with a cloth like this. Once it's ready, you remove it. Along with that, you have a standard which is not used in the autoclave to compare your autoclave. Then break it. There will be an ampule-like thing within the thing, that is the broth used, and incubate for 48 hours to see if there is color change. There are quick readers which are available now, which will get the result within 3 to 4 hours. And have any of you seen what is really inside a biological indicator? It's got a cap with a hole for the steam to penetrate, and there will be a filter, paper filter, to filter any other bacteria or spores entering the biological indicator. And the broth is there, which we use for the spores to generate if it is live. This is the broth within it, and ultimately this is that small disc what you see is the 1 million spores. Okay, that is the indicator. Then different spores are used for different sterilization. This I had already shown you. And steam and formaldehyde should not be used in ophthalmic practice. There are indicators for it. Definitely Cydex should not be used for ophthalmic use. For ETO we have different sterilization indicators. One is the biological, which I had shown already. For plasma sterilization, the indicators, there again the biological indicators can be used. This is the plasma, and these are the indicators for it. Thank you very much for your patience.